So I'd like to just take the next few minutes to really think about what we need to learn in order to develop disease-modifying therapies that can alter disease progression and hopefully eventually interrupt disease emergence in inherited cardiomyopathies. So we need to understand how gene mutations actually lead to disease by better characterizing the phenotypic spectrum and disease mechanisms of genetic cardiomyopathies. And fundamentally, we need to understand why some patients do poorly while others do so well. And so we can identify key patient subcohorts and really understand which patients we want to target aggressively for medical therapy. And then how can disease progression and emergence be prevented? And so by understanding the genetic basis of disease, we really have an incredible opportunity to provide disease-modifying therapies at a time when they might be most effective, at a time when disease risk is known to be present because of the presence of an underlying pathogenic gene mutation, but the overt manifestations of disease and entrenched manifestations have not yet developed. That might be a time biologically where it is easiest to act. And so, for example, with hypertrophic cardiomyopathy, we know that the mutation that actually is responsible for causing disease is introduced at the time of fertilization or shortly thereafter, and is certainly present at birth, but it can take years and decades before demonstrable left ventricular hypertrophy can be detected and a clinical diagnosis of HCM can be made. Typically, this isn't until adolescence or even earlier middle adulthood, although certainly there's childhood onset disease. So can we understand more clearly how sarcomere mutations actually lead to hypertrophic cardiomyopathy? And can we identify early phenotypic changes that can isolate the changes that are caused directly as a result of the mutation from the secondary consequences of remodeling and adverse effects caused by disease itself? And then how can we use this information to really understand how disease progression can be interrupted? So it's, and if you think of it this way, it's really a remarkable opportunity in medicine because it's not so often that we have the ability to identify with high precision and accuracy people that are at risk for developing disease far in advance of when disease might actually develop. And so there are a couple of different models that we can think of about how sarcomere mutations actually cause HCM. It seems that so initially, some of the fundamental effects of the mutations, based on animal models and basic models in vitro, seem to be increased ATP hydrolysis, increased sliding velocities, and increased calcium sensitivity, all leading to increased force generation, again, as a direct result of the sarcomere mutation itself in isolation. So this increased force can be sensed by the non-myocyte or fibroblast population resident within the myocardium with the activation of TGF-beta, further activation of fibroblasts, and elaboration of profibrotic molecules, causing eventually fibrosis, both interstitial fibrosis and dense replacement fibrosis. So there are a couple points along this pathway that we might think about targeting for disease-modifying therapy. We can try to target the increased force directly with allosteric inhibitors of myosin activation. We can think about trying to alter calcium sensitivity with agents that might impact intracellular calcium handling, such as diltiazem. And we can think about trying to inhibit TGF-beta activation using angiotensin receptor blockers. So all of these strategies have been successful in mouse models, but can we actually successfully translate them to human management? So there are clinical trials that they're ongoing now that I'd like to introduce, and also introduce some of the basic science and the early mouse trials that support their use. So first, I'd like to talk about this molecule developed by MyoKardia, a small startup biopharmaceutical company trying to target different components of the sarcomere. So this compound MYK-461, which has recently been named mavacamten, was developed to reduce ATPase hydrolysis and force generation in the myocyte. And so it actually inhibits the ATP hydrolysis here in this initial rate-limiting step of the chemomechanical cycle of the sarcomere. So it's the initial step of ATP hydrolysis that's required for the thick filament to bind to actin to lead to the power stroke, where there's, again, another round of ATP hydrolysis to allow for the detachment of the thick and thin filaments to start the cycle all over. So 461 actually interferes, again, with this first rate-limiting step. And you can see that it does decrease the rate of ATPase hydrolysis in isolated myofibrils. And similarly, in isolated cardiomyocytes, it does decrease fractional shortening in a dose-dependent manner. And so when given to mouse models of HCM, these are mice that have different mutations in myosin heavy chain that ultimately cause a phenotype which closely replicates that of human disease. Mice that are untreated, so this is a mouse that has the myosin missense mutation, arginine 403 glutamine. So in the untreated state, it develops hypertrophy around 20 weeks of life or thereabouts. But when treated in the pre-hypertrophic phase with 461, you can see that there's attenuation in the amount of hypertrophy that develops into levels that are very similar to the wild type naive and the wild type treated animals. And we can see this also with a reduction of fibrosis. Again, these are mice that are treated in the pre-hypertrophic or preclinical stage. There's a decrease in the amount of hypertrophy they develop, and also a decrease in the amount of fibrosis that's developed. So here's the untreated mouse. You can see all these blue areas show as represent collagen in the trichome stain. There's much less collagen grossly, and also if it's formally quantitated with 461 treatment. And so MyoKardia has recently completed phase 1 and phase 2 trials giving mavacamten to human populations with HCM. So these are small scale trials. These represent a total experience of about 20 individuals. You can see that mavacamten reduces LVOT gradients and also increases functional capacity and increases peak VO2 after 12 weeks of treatment. The effects are seen pretty quickly, and then these are showing peak VO2 changes from at baseline and then at week 12. So they are now in the process of starting the pivotal trial for obstructive HCM, which is called Explorer. I think that both Explorer and Maverick, which is an exploratory trial for non-obstructive HCM, have dosed their first patients now. And then there's also been, there was early excitement and success in mouse models using agents that interfere with intracellular calcium handling, including the L-type calcium channel blocker diltiazem. And so, if mice were treated in the preclinical stage, there was less development of hypertrophy and less development of fibrosis than in untreated animals. You can see that it wasn't a complete cure. There was still significantly more hypertrophy that developed even with diltiazem compared to wild type, but that was substantially reduced compared to what would happen if there was no treatment. And so treatment was most beneficial when, again, it was given in the early preclinical stage before LVH developed. If they actually waited and started dosing mice after they were allowed to develop a full phenotype, there wasn't any obvious benefit. Diltiazem was not able to rescue or reverse established disease. And there were exciting clinical implications. This was actually the first time that pharmacological therapy was used with the intention of changing the natural history of HCM. And it showed that just giving a pill or a drug, an oral drug, that improved calcium balance in HCM may actually improve the natural history. So with this in mind, we performed a very small pilot trial of disease modification using diltiazem versus placebo, again, in preclinical humans with HCM. So these were individuals that carried a sarcomere mutation that was the cause of HCM in their families, but have not yet developed a clinical diagnosis of HCM. They were stratified by age and then randomized to receive either diltiazem. So there were 18 individuals that received diltiazem with a mean age of 14, and there were sorry, this didn't show up, but there's 20 individuals that received placebo at a mean age of 17, and they were treated anywhere from 1 year to 3 years, depending on when they actually enrolled in this study. We followed many different parameters, echo, MRI, and biomarkers. And so as an initial just pilot experience with this, it was really testing safety and feasibility. We wanted to figure out if we could give medications safely and reliable to young, otherwise healthy and asymptomatic individuals. The last thing that we wanted to do was to take a young person that was feeling fine and give them a medicine that might interfere with either school performance or daily functioning. And then we wanted to see, can a treatment response actually be detected? So I think that this is actually the most challenging part of trying to do these disease-modifying therapies, because it's relatively easy to show that you've made a very sick person a little bit better, but to try to show that you're making, a person that looks, pretty well a little bit better is a much greater challenge, especially when we're dealing with relatively rare diseases where study sizes will be quite small, and we're talking about a disease whose natural history plays out over decades, not over the usual 1 or 2 years that we typically have available for a clinical trial. And so as a pilot study, we studied many, many, many different parameters and largely didn't see a significant difference between those treated with diltiazem and those treated with placebo over the period of study. But one result did catch our eye, and that was the effect on left ventricular cavity size. So sarcomere mutation carriers, even when they have normal LV wall thickness, tend to have smaller cavity size, and that's represented by having a z-score that's less than 0, meaning a z-score that's below the population mean. And indeed, at the start of the trial, the subjects had a z-score of about negative 1.3 or thereabouts, meaning they were a little over 1 standard deviation below the population mean in terms of LV cavity size. And so when treated with diltiazem, you see that there was an increase in LV cavity size towards population mean during the period of treatment. And then when we followed them up about a year after treatment, that value had largely returned to what we had seen at baseline. But in placebo, in the placebo-treated individuals, there was just a continued decline in LV cavity size. And so this suggests that there might have been some impact on left ventricular remodeling, and furthermore, outside of the effect on LV cavity size, there might be other effects that we might expect to see related to filling, because there's also a decrease in the thickness to diameter ratio. If the wall thickness isn't really changing substantially, but the LV cavity size is getting a little bit larger, the thickness to diameter ratio will decrease. And if we have a decrease in the thickness to diameter ratio, diastolic wall stress will consequently increase, and therefore lower diastolic pressures are needed to fill the left ventricle. So these purely geometric effects can actually influence diastolic filling in a favorable manner. And then we looked also at whether the background genotype might also play into differential effects. And we found that patients with myosin-binding protein C mutations seem to have a more prominent treatment benefit. Okay, so take in mind that these are very small numbers, and so this is really completely exploratory and hypothesis generating. But those individuals with myosin-binding protein C mutations that were treated with diltiazem actually had kind of stabilization of their maximum wall thickness during the time of treatment, and then an increase when they were followed up after treatment. But those with myosin-binding protein C mutations treated with placebo and myosin heavy chain mutation carriers treated with either diltiazem or placebo just had an increase of LV wall thickness. As similar beneficial patterns were seen in the myosin-binding protein C mutation carriers with respect to LV mass on CMR, serum troponin levels, and E over E prime ratios. So this suggests that the response to disease-modifying treatment may indeed vary based on genetic background. And I don't think any of us would necessarily be surprised about that, but it's just it shows us another layer of complexity that we need to take into account when thinking about these already heterogeneous populations. The background genetics may indeed matter quite a bit. And then finally, we've been looking at whether we can exert disease modification through inhibition of TGF-beta, since activation of TGF-beta seems to be an early step in the pathophysiologic development of hypertrophic cardiomyopathy. So again, if animals are untreated, in the face of an underlying sarcomere mutation, you can see that lots of fibrosis develops with the onset of phenotype. But in animals that were treated either with a TGF-beta neutralizing antibody or with the angiotensin receptor losartan, had much less development, almost no development of fibrosis. And so the idea of using angiotensin receptor blockers in HCM has been explored a number of times. The largest study was performed a couple of years ago by our colleague Anna Axelson, an incredibly talented cardiology fellow in Copenhagen. She randomized 133 patients to either losartan 100 milligrams or placebo and treated them for 12 months and was looking for a primary endpoint of a change in LV mass. And you're using that as the bar. There wasn't any significant difference in patients that were either treated with placebo or with losartan. There was a mean difference of just about a gram per meter squared over the treatment period, and that was very similar in both of the treatment groups. There also wasn't a significant change in any of the secondary endpoints that they looked at. And so, why might this be? So again, they found that there was, no significant change of losartan on LV mass or the secondary endpoints in patients with overt HCM. So the population that they enrolled was an average age of 52. About a third had symptoms of heart failure. But they also did not have any adverse events. They actually randomized patients that had obstructive physiology to receive angiotensin receptor blocker and found that those patients did just fine. So that suggests that maybe we can use ARBs if we are really stuck with patients with refractory hypertension and if initiated very carefully. And so where does this point us? Is this just the end for ARB therapy in HCM? Or, might we think about some of the other reasons that this study might have been potentially negative? Were they targeting the right patients? Based on the animal studies, it seems like we really want to act earlier in disease, ideally at a preclinical stage. But, even just earlier in disease, there's a long ramp-up time between when a diagnosis is made and when serious symptoms and adverse consequences develop in HCM. So it may give us a bigger window of opportunity to treat. So do we want to treat younger patients with earlier on in their disease course where they might have more milder disease and more reversible manifestations? And do we want to specifically target sarcomere mutation carriers? Because again, all of the preclinical data supporting the role of TGF-beta activation was on sarcomere mutation carrier animals. And are we looking at the right outcomes? Might we want to look at outcomes that are much more dynamic and responsive than perhaps change in LV mass over a short period of time? So with this background, we've been involved in the VANISH trial, which is a phase 2 randomized placebo-controlled double-blind clinical trial using valsartan, a somewhat more potent ARB in terms of effects on TGF-beta inhibition in early sarcomeric HCM. So we're targeting a younger group of patients with less hypertrophy and no symptoms, again, randomizing to valsartan or placebo for treatment of 2 years. And our primary endpoint is a composite looking at change from baseline in different domains of variables that reflect myocardial injury, hemodynamic stress, collagen metabolism, and functional capacity. So we just completed randomization about a year ago. We have 178 patients in the primary analysis cohort, and we also have about 38 patients in an exploratory arm, which is looking at mutation carriers with normal LV wall thickness, but with some phenotypic manifestation that the sarcomere mutation is doing something, either abnormal EKGs, low E prime velocities, or increased thickness to dimension ratios. And so our follow-up will be completed in about a year, so stay tuned for those results. And then finally, disease-modifying therapy is not confined just to hypertrophic cardiomyopathy, but also extends to other genetic cardiomyopathy. And I think some of the most exciting research deals with Lamin cardiomyopathies. So Lamin encodes this protein lamin AC, which is an internuclear membrane protein, which provides structural integrity for the nucleus and also plays a role in mechanical transduction and gene expression. So there's a very well-described cardiomyopathy that is associated with dominantly inherited mutations in lamin. It's highly penetrant. Heart block and atrial fibrillation tend to develop earlier in life, usually before overt DCM develops. Patients tend to do worse in the presence of a lamin mutation than all-cause dilated cardiomyopathy, with a much greater risk for cardiac arrest, heart failure that's refractory, and also stroke. And targeted therapies are being studied. Particularly Array Biopharma has developed some targeted therapies for lamin heart disease based on the observation that activation of the P38 MAP kinase pathway has been established to be important in the early pathogenesis of lamin cardiomyopathy in mouse models. And so they developed a small molecule inhibitor that seems to favorably influence disease natural history. When given to young mice, you can see that there's an improvement in fractional shortening and a decrease in LV cavity size. And so there's a phase 2 trial that was recently done in a small number of patients with lamin cardiomyopathy. And they showed that patients treated in this manner had improved 6-minute walk times, decreased BNP levels, and stabilization of their EF. And so now a phase 3 clinical trial is pending and about to start enrolling. And then finally, since we're talking about genetic heart disease, instead of using pharmacologic therapy to try to counteract the downstream effects, can we actually think about either silencing or editing the mutation itself? So a few years ago, gene silencing techniques using interfering RNA were performed on mouse models of myosin heavy chain HCM, hypertrophic cardiomyopathy. And so injecting a short fragment of interfering RNA at day 1 of life was able to decrease expression of the mutant allele by about 30%. And this prevented the development of HCM in these mouse models. So that was interesting in that you didn't have to completely knock down the expression of the mutant allele. Even just decreasing expression by 30% seemed to prevent the development of HCM. Of course, the durability of this one-time treatment is unclear. And then most recently, there was a proof of concept study that was published in Nature showing genome editing with the CRISPR-Cas system. And so they were able to do germline correction of a heterozygous MYBPC3 mutation in zygotes, so really just immediately after fertilization, and show that they were able to develop embryos which were free of that mutation. So just in summary, again, this shows us the power of collaborative basic science and clinical and translational studies to really help us understand more fully the molecular basis of genetic cardiomyopathies and how disease expression occurs in humans. And these mechanistic insights can really help to transform our management, moving us from a palliative stance to a much more proactive stance, developing rational mechanism-based therapy to modify disease and to identify and target the highest risk cohorts before irreversible changes occur. All right, so this is the team back at home and all of our collaborators, and thank you very much for your attention.